Rolling?
Yes, sir.
All right, good deal. So the pblu lab, start off like right there with that. This is the lab we're going to be doing on Monday. Why am I recording this? A lot of people are gone. You guys can review this if you need to. This is the meat and potatoes of what the heck's going on. So we've done a unit on genetic technology. We're going to do DNA transformation. Okay? We're going to transform bacteria. If you can't read what that says because I have it pulled down too far, I do it every day, right? Transformation, we know what that is, right? Anybody know? Remember?
Yes. It's like where you add DNA to make a different thing.
Where? It's, yeah, bacteria, right?
Yeah.
Specific to bacteria. Very good.
Insulin.
So, yeah, and for our purposes, when I just said know that we're talking about transformation, we're talking about in bacteria specifically. Okay? We're going to be using E. coli. On the test, that was a question. E. coli is the most commonly used vector, or the plasmid is using that, alright? So, we're going to have two foreign DNA genes. One's for antibiotic resistance, and one's for an enzyme that break down lactose. We'll get back to this. This is complicated stuff. That's the hard parts of that. Why are we doing this? It's cool and it's useful. Obviously, I'm going to say it's cool. Transformations are used in many lab experiments. Remember my idea with insulin we talked about, right? We talked about the diabetes, right? So this is used to synthetically produce it, right? Because it's an epidemic we have in this country. Not enough, not identically structured. On your test, it was like you could have an allergic reaction if you got insulin from a pig or a horse. Pig or horse or cow or something like that. Some type of mammal with 4 legs that we used to get insulin from. Alright? So we put it back in the human. So instead of getting it that way, obviously we make it synthetically now through transformation. So step 1, these are all little plasmids. I know this is kind of this is a prokaryote, right? Because this is not the nucleus, right? This is that one chromosome that attaches to the cell membrane. Okay? So the plasmid. And we're going to get something with an antibiotic resistant gene. What does antibiotic mean? Does anybody know? Antibiotic.
It's resistant to biotics.
What's a biotic? Yeah, you're right. You're right. It is.
Resistant to living things.
It kills. Yeah, good way to put that together. It kills bacteria. Antibiotic. Bacteria killer. Everybody hear that? And you said it, and we got it, so we're good. All right. So basically what we're going to do is we're going to take out this plasmid from this E. coli right here. And what do we need to do to it next, you think?
Insert a gene.
How are we going to do that?
Restriction enzymes.
Ah, restriction enzymes. We're going to cut it. We're going to cut it with restriction enzymes, right? We're going to get some sticky ends, right? We're going to get some hydrogen bond. We're going to get some DNA ligase. We're going to get some, yes, reincumbent DNA. So what we're going to do is we insert the modified plasmids. We're going to insert a modified plasmid. Okay? So, imagine I have an antibacteria right here and the food source. It's going to kill, this is important, it's going to kill everything that doesn't have this little plasmid here. Okay? I don't know if you see this over here. This is you guys see that right there? How that guy has it too? Everybody seeing that? Yeah. Alright, good, because it kind of cuts it off and it's confusing. So notice, this has a gene that's going to not let the antibiotics kill it. It's going to produce a gene, which is a polypeptide eventually, that's going to protect it from the antibiotic, the bacteria killer, right? So what's going to happen is, if I put this in this, these are going to die, okay? Those are going to die. Only the ones with the new plasmid now are able to live in this environment. Does that make sense? Yes? Follow me? So, because a lot of times we're gonna put plasmid here. We can't get it. We don't know. We're doing this. This is called the shotgun approach. We're putting plasmids in hopefully a lot of these bacteria cells. Are we gonna be able to put it in all of them? No, just by sheer chance. We're just doing sheer numbers here. Okay, it's kind of like throwing. Okay, let's use a shotgun in this analogy. I've never said this before, so it may be crappy. So, imagine throwing confetti up in the air. Right? Those are bacteria cells. You have very, very tiny shotgun pellets, right? You just take it, boom, okay? Whichever one confetti gets hit with the shotgun pellets, those are the ones that got in there. You're not all going to hit. Yes? I don't know if that was actually decent. Okay?
Now I want to shoot confetti.
Now, yeah, how's he say it?
Diabetes.
Diabetes, yes. So now we can collect and purify the insulin and help use people with diabetes. So like here it says, publicly mispronouncing diabetes since 1999, right? So anyway, yeah. Pretty funny. So this is You Are the Future. It's kind of cheesy, right? Yeah, so I got this from one of my really good friends who's also a biology teacher in Columbia, Missouri. So her and I kind of think similarly. So anyway, that's where I got this from. All right, that's that. Okay, keep rolling. Okay, I'm just going to kind of fill time right now, right? Now, that was just an example. Hopefully that wasn't mind-blowing, right? That wasn't like, what is he talking about? I have no idea what Mr. Smith is saying. It's all gibberish, blah, 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 blah. All right? Still buying time. Seeing that? All right, so, now, could I? All right, let's see. Let's go this way. Zoom this. Uh-oh. Is that fitting in there? This is technical difficulties. It's on me. It used to be not, and my battery's at 17%. That's awesome. Give me a couple seconds. Technical difficulties. So let's need to be playing a tune like that. We're currently having technical difficulties.
I don't.
Jeopardy.
What? Yeah, that's what I was
No, it's like it really didn't happen anymore. Okay, that's what I'm looking for.
It's like the hold.
Do this.
Like elevator music.
Like,
Let's zoom out.
Do, 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 do.
that's Jeopardy.
All right.
That's not elevator music.
So,
It's time buying.
good. This is all we're just recording me on. That's good. Watch me sweat. Now. Alright, now, what we're going to do, so these are my notes that I'm going to share with you, okay? This is stuff that's all on Moodle, under genetic technology. If you looked at today's bell ringer, it said to do what to this?
Download it.
Download that, right? If you're not doing it, follow the directions. Shame on you, okay? So, there's this stuff called LB, Luria broth. You know, I probably mispronounced that, whatever. So, what my old friend that kind of helped create with this lab, she called this chicken noodle soup for bacteria. Basically, bacteria love this and they grow like crazy on it. Okay, it's like a nutrient broth. Everything they need to grow up big and strong and produce baby bacteria. Okay, so it cannot grow in the presence of ampicillin. You guys heard of penicillin? Right? Similar. It's an antibiotic. Remember what's an antibiotic?
It kills things that don't have its plasmid.
Even simply put, it's a bacteria killer. Keep it gangster with an A, okay? Alright, so, now it's bacteria killer. Killer, alright? So, now. So, normal stuff can't grow in ampicillin, okay? So, amp, what do I have here? Amp is a form of, and we're going to call that amp. It's ampicillin. Ampicillin is a type of antibiotic. All right? Ampicillin is a form of an antibiotic. There's a bunch of different types. Okay? So that's basically antibiotic is something that's, if I remember correctly, it's like it's a form of a protein. Okay? It's a protein, obviously. So now we're going to talk about this stuff called, how am I going to say this? So notice this has plasmids in it, but it doesn't have the plasmid that we want. Okay? It doesn't have the plasmid that we want. Alright? What we're going to do is, yesterday I got in the mail, it's in the freezer right now, I got plasmids with the right stuff already. I've already got recombinant DNA plasmids. It's in the freezer right over there. Okay? If time allots at the end of the hour, I'll show you the plasmid, and I'll show you what the E. coli looks like strained. So this weekend I have to make all the E. coli, all the plates for you, and get them growing. Okay? And get them going. Alright, so, but anyway. So, what we're going to do is Gene A makes, this is going to be confusing for a second, but hang on, okay? So remember, this is going to go into there. That's what we're trying to do. We're trying to get this plasmid, this pblu plasmid, into here. That's the whole purpose of this, okay? So we're going to try to do that. Alright? I'm going to scroll down for a second. It's not a deal situation. I got to get stuff. Stop it. Doing dual monitors. So we're going to try to get that into this here. This is probably this part of this is the most beneficial. Okay? So if I have this in here, this should be a B there, okay? Remember, gene B is like a section of DNA that codes for a protein or polypeptide chain. Makes beta-galactosidase. It's an enzyme that breaks down lactose. What's lactose?
It's found in milk and it's a sugar.
It's a sugar. How do you know it's a sugar?
Oats.
Ends in os, right? Good. So there you go. So, it's a chemical. So, now what I'm going to show you now is that AMP Mary, are we getting these in the... Are these actually showing up in my writing? Can you just see them? Okay. Amp and X-gal are chemicals, or some type of chemicals. I'm going to call them chemicals for now to make it simple. They're more complicated. Chemicals that we put on auger plates. I'll explain what auger plates are in a second. Okay? So amp and X-gal are chemicals that we put on. Ampicillin is an antibiotic. This is actually something that... Okay. So this is actually something that we're going to put in here. So actually if I put this, so if I have this plasma in here, it's going to be able to live in the amp plate. So I'll explain this in a second. If it has this and this, okay, if it has both these in here, it'll turn blue. So that's why it's called the pblu lab. All right, and I have a picture of this up here. I'm actually going to tell me if I'm in frame. Do you see this?
It's too dark.
Is it too dark?
Yeah.
Okay, well, I would show the Googles. How about this? Is it better?
Yeah.
So that's what it should look like at the very end. Okay, you guys see this kind of? And that little thing it's growing on, do you remember from Alltech? That is like a transformation plate. That lady that had the auger plates that was looking at it, that's what we're going to be doing. Okay? That's what we're going to be doing with this.
Wait, is an auger plate the same thing as a petri dish?
Yes.
Okay.
The auger is the auger, and the auger is like the growth medium. Okay? It is. It's a petri dish. Okay. Yes. Good. It's the exact same thing. Now... So what we're going to do is, guys, check this out. I'm going to zip through this quickly because we can watch this again, but you can watch this again. This is going to be my plasmid, okay? I'm going to take my plasmid. No. You're going to take my stuff. No. That's a coli. What are you doing, Mr. Smith? Trying to mess him up already? Starter plate. This is my starter plate, okay? These little dots, what these little dots represent, this is the, I have, okay, never mind, I'm not going to say that loud. These little dots are little E. coli colonies. Did you guys hear that? So what I'm going to do this weekend is I'm going to make some I'm going to take some E. coli from the fridge, put them on some cultures. We're going to start growing that E. coli. Okay? These are going to be little pieces of E. coli. You're going to scrape off some of that E. coli. You're going to have millions of cells. Okay? And you're going to put that on this little loop that's going to look like this. It's real tiny. Wait, let me go over here so you can see it. And yes, laugh at my drawing skills. Okay? Okay? It's going to have a little inoculation loop. Think of it like, you know how you blow bubbles, but real tiny. Got it? Alright, good. Now, so we're going to take that, we're going to put this into here. So now I have millions of cells in there that don't have the plasmid, right? Or nucleus. Alright, yes. So then what we're going to do is we're going to put them in cold calcium chloride. CaCl2. Okay? You guys learning the name ionic compound yet?
Yep.
Yep, CaCl2, Ca plus 2, Cl minus 1, crisscross applesauce, and there you go. So, alright, now, so we're going to do what the calcium chloride does, if I remember correctly. No, we won't worry about that yet. Don't worry about that. Okay? So notice, one of these is the plus. This one gets the plasmid. See this little thing here represents? Number 2, this is representing the one that gets the plasma. This is the one that doesn't get the plasma. Why would I squish? I would do this squish, right? No. Why would I do that here as I would do this here? This is a pipette, by the way. Remember pipettes? From messing with Daphne back in the day? Yeah. Alright, so nice. Yeah. Right, so boom, boom. So why would I do this on the other one?
Kind of keep it from settling.
What's the word I'm looking for? Looking for a word in experimental design I'm looking for.
Oh, not constant.
Constant!
Constant?
Constant's perfect. That's a constant. Okay? That's a constant. But this would also be our blank in this group here eventually. Control. Very good. Good. So, not constants, exactly constants. Alright, so.
Okay.
No, it's good. So now what I'm going to do, so one has a plasma, one does not have a plasma. This is doing our shotgun technique. Okay? So now what you're going to do, so what the calcium chloride does, and the hot water do together, you're going to put it in hot water for 90 seconds, exactly 90 seconds. That's going to make, basically, the cell membrane just kind of deteriorate momentarily. Because we know that phospholipid bilayer is a fluid thing, right? So if we heat it up real quick, it can kind of go away, it can kind of put gaps in it. That gap's big enough for that plasmid to go in. And then as soon as we get done heating it, we immediately, this is important, you immediately go from here to here and put it in ice cold water. So what's that going to do?
Close it.
Close it back up, shrink back up. Kinetic molecular theory that you'll learn later, right? So yeah, how hot stuff is, how fast the molecules are moving, right? So, good. So this is what we're doing. This is kind of a broad overview, and this is a much better explanation, this picture diagram here, than what this actually kit that it came with says. Excuse me. So you're going to allow in the room for 10 minutes. Then what we're going to do, so this is also another important part. Same pipette. Here's some sterility issues. Don't worry, I'll go over this. Plus LB means it has it's luria broth. What's luria broth again? Chicken soup for the bacteria soul. Okay? Oh, it's cheesy. All right, so now LB plus amp. So this means this plate just has LB in there. Okay? This plate has LB and amp. What's amp again? Ampicillin. So it's going to kill all the bacteria that don't have plasmids that we inserted, the new plasmids, right? Good. But so this one's going to have LB, amp, and X-gal. So remember, these are two chemicals that are in there. These are nutrients, right? All that's going to be in there, right? So this is going to. So this one is going to turn blue. Okay, because the X-gal reacts, if you go back and look, X-gal is, what's the word here for it? Let me show you. 5-bromo-4-chloro-3-indole-beta-delta galactosidase. So instead of saying that, they just call it X-gal. You guys okay with that, just calling it X-gal? Yeah. It's a long, complicated chemical thing. Basically what happens is when you have that plasmid in there, in the presence of X-gal, it's going to, for our purposes, magically turn blue. Everybody with me? If you want to get into the depths of the biochemistry behind that, come see me before or after school, and I'll try to explain it. Okay? So these are different levels of independent variables. You see it? Boom, 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 right? Don't have that, don't have that, don't have that, okay? What no? Let's see. Make sure I didn't say that right. Yeah, okay. No, I'm sorry guys, I screwed that up. We're just going to have 3 of these here, okay? So let me make sure I am telling you guys correctly about this. Okay? So I wasn't expecting doing this today, but it just kind of worked out well. I had time to actually look over this stuff. So you're basically going to do this, I believe. Oh, you see the plus? What does the plus stand for? I'm sorry, guys. Ignorant of me. This means it has the plasmid. Does that make sense? Plus the plasmid, has the plasmid, has the plasmid. Then we're going to do these that don't have the plasmid, don't have the plasmid, don't have the plasmid. Does that make sense? So these should all have growth. This should have growth, this should have growth. All of these should have growth. This should have growth. None of these should have growth. We doing okay? There are some sterility? Is that the word I'm looking for? There's some issues with this. This lab will not work if you're in a rush, if you're hurrying. That's why we're doing a double block. Okay? So do not put down your pipette or inoculation loop. There are sources of contaminants. Every year I've done this, I've had students that are successful with this. Okay? With getting the stuff to grow like it should. There'll be a grade on getting that done. If it doesn't happen, is it going to make you fail the lab? No. But I think that people should be rewarded. It's going to be like a couple points. It's not going to be like, it's not going to be like, oh my gosh, I have an F now. No. But I want to reward the people and give rewards for people that are doing these things well. Okay? I will explain more of the sterility stuff after we get done. On Monday, excuse me. We'll go through that real quick because I think that's important. And I'm going to kind of explain a couple more things on the video before I stop talking. Okay? I think that's pretty much. So there's a 4-person. There's 4 people in a group. Okay? There's a job for each person. Like, what are our lab groups? I'm going to create them this weekend. Okay? I'm going to create them this weekend. There will also be a lab grade on how well prepared you were, or know what the heck you're doing, and if you're working on something, or if you're just letting somebody else do all the work. Okay, so I would look at that as an easy point. Okay, all right, that's an easy point that can help your grade if you struggle with other parts of this. Okay, so let's say you don't get it done, you don't get the lab to work, but you worked hard and the other stuff, that would take place of that grade. Okay, that one little grade. Does that make sense? So I want you guys to work hard on this. All right. The last thing, and these are all the sterility things. All of this is on where? Moodle. All right. Now I'm going to show you some stuff. So.
Is that necessary right now?
Is it still rolling?
Yes, sir.
Yes, good.
We're having a feature of students right now.
Yeah. That happened. It happened on camera. Roll with it.
I'm surprised it stayed up that long.
All right, I'm going to turn on the lights for a second. Okay. This is non-harmful strand of bacteria. Let me be clear about that, all right? That being said, don't do idiotic things in the lab. Okay? I'm not calling anybody an idiot. I'm just saying don't do idiotic things in the lab, right? Now, this is the reincumbent DNA in here. There's a lot of them. This is why this lab is expensive, because of this plasmid here. This is the plasmid that has the 2 genes that we need right here. Okay. Other stuff you don't care about. This is the cool stuff. Okay. This is a. Am I supposed to do anything with this? Am I supposed to open this? Am I supposed to refrigerate on a line? Am I supposed to keep this open? I don't think so. Okay, no. All right, so maybe. Thought like, oh, it's bacteria, it needs oxygen. Okay, anyway, never mind. But anyway, this is the E. coli, they call it the colony. So I'm going to take from this, the little inoculation loops, those little bitty guys, I'm going to scrape down in there, and I'm going to put that on these plates here. Okay, these are the plates that we were talking about. I'm not going to take these out, okay? Then I'm going to grow this strand of E. coli on these plates for you guys. I think I get them started for you, okay? So I will grow them for you. All right. So all you have to do is you have to what's it called? You just got to put the plasma in there and do the other procedures. Okay. This may be tweaked slightly once I get into the get here on Sunday and start working on this, and we may have to change things a little bit, but this is what I'll be doing. Okay. Everybody, is that okay? Everybody cool with this? Yes. This is one of the coolest labs. That I am a part of. I'm a part of. I like doing this. All right, so this is a fun one. It's labor intensive. So on Monday when you come in, there'll be stations set up. There'll be a hot water bath. That flip the breaker if I put a hot water bath back there? No. Okay. There'll be a hot water bath. There'll be an ice station. There'll be a station for there'll be a station set up. Okay. How many stations are there? Does it say how many stations there are? No, but I'll have them. There'll be 2 or 3 stations. I'm going to use it in here because I think it's easier with the lab space. Okay. I think we're good. You can go ahead and stop recording.